Okay, good afternoon, everyone. You can hear me? Okay. It's my great pleasure to introduce Dr. Nisha De Silva, BDS, MSD, PhD, who is the Donald Kerr Endowed Collegiate Professor and Associate Chair of the Division of Oral Medicine and Pathology and Radiology at the University of Michigan, a School of Dentistry. She's also Professor of Pathology at the Medical School and a member of the Rogel Cancer Center. Dr. De Silva received her BDS degree from the Bombay University of Bombay, India, followed by a residency in oral and maxillofacial pathology and a master's degree from Indiana University. Then she followed that with a PhD from University of Washington, where she got her PhD degree, and then later joined University of Michigan as a faculty. She received a Distinguished Scientist Award for Oral Medicine and Pathology Research from the International Association of Dental Research, and her work is highly recognized across the medical community. Dr. De Silva is a dentist scientist, a translational research in oral cancer, focusing on biomarkers and molecular mechanisms of tumor progression and treatment resistance. She has won many accolades, among which I'm just listing a few. Dr. De Silva is a diplomat of the American Board of Oral and Maxillofacial Pathology, which is her area of clinical practice as well. She enjoys teaching and mentoring, and a recipient of Michigan Institute for Clinical and Health Research Distinguished Mentor Award. She also serves as a member of the National Advisory Dental and Craniofacial Research Council. Her work has been funded by National Institute of Health and currently by the NIH, National Institute of Dental and Craniofacial Research, sustaining outstanding achievements in research award, and the NIH and NCI Cancer Institute's grants. With that, I invite Dr. De Silva to share her research with us today. Thank you.
Thanks, Dr. Nagrath, for that kind introduction. It is a pleasure to be here today to talk about our work in oral cancer. And on our screen here, I have a picture of a clinical presentation of oral cancer, this reddish patch on the posterior floor of the mouth and ventral tongue. If you take a section through this patch, what one sees is essentially the epithelium that lines our oral cavity. And in that epithelium, the blue cells depict what we call transformed cells. As long as these cells are retained within the surface epithelium, it is considered a precancerous lesion. But once they breach this basement membrane and they invade the underlying tissues, these transformed cells are now malignant, and this condition with the tumor islands in the underlying tissue is essentially squamous cell carcinoma. From the underlying tissues, the tumor cells can spread to adjacent sites, and most significantly, they can enter the vasculature or the nerves and spread to distant sites. So this process of invasion is very critical in both tumor development and spread. And today I'm going to talk about our work in one aspect of this, and that is perineural invasion. Before I introduce perineural invasion, I briefly want to give you an introduction to head and neck cancer. There are about 800,000 new cases of head and neck cancer each year across the globe. 90% of these are squamous cell carcinomas. So you'll hear me use the term squamous cell carcinoma interchangeably with oral cancer. And the 5-year survival rate for this disease is pretty poor at just 50 to 60%. Now, in large part, this poor 5-year survival rate is due to late detection. Here's an example of a lesion that looks pretty innocuous clinically and is in an area that is hard to visualize, meaning the posterior floor of the mouth. And often these, at this stage, these lesions can be asymptomatic. So that contributes to late detection. The treatment for this disease is pretty debilitating. It's either surgery alone or a combination of surgery, radiation, and chemotherapy. And unfortunately, the treatment is selected by tumor stage, and early stage lesions such as stage 1 or stage 2 would get surgery alone. Late stage lesions such as 3 and 4 could be treated with a combination of surgery, radiation, and chemotherapy. The presence of lymph node involvement upstages the lesion, and then those patients would get aggressive treatment. Perineural invasion, which is cancer in the nerves, is often used as a surrogate marker for lymph node involvement. And I say surrogate marker because it is not always possible to detect lymph node involvement clinically, because one may have what are called occult, meaning detected only on sectioning the lymph nodes. So perineural invasion may be used to as a surrogate marker for that. So what is perineural invasion? Perineural invasion is basically detected in multiple cancers. It's not specific for oral cancer. It's seen in pancreatic cancer, prostate cancer, gastric cancer, etc. And its presence is considered an adverse risk predictor. It is a challenge to eradicate. As you can imagine, if you look at this innervation of the head and neck region, these nerves travel throughout the face, and some of them are very fine. And once the cancer gets into the nerves, it can spread to regional and to distant sites, even into the brain. And these patients tend to have poor survival. Initially, the patients may be asymptomatic, but later they may develop sensory disturbances. For example, they may have extreme pain, or they may have paresthesia, similar to what one would have if you went to the dentist and got a anesthesia for a tooth filling, but then the anesthesia just persists. So in the case of cancer, the anesthesia could just persist, and we call that paresthesia. When PNI occurs, the treatment is very aggressive. Now, basically, at a histologic level, the way one defines perineural invasion is this presence of tumor cells surrounding the nerve completely. On the right are sections from patient specimens, and on the left is a caricature to help you visualize the tumor surrounding the nerve. Or the tumor can surround up to 33% or lie in close proximity or within the nerve. So all these are perineural invasion. This was considered a very passive process, so on the part of the nerve. So the nerve just kind of sat there, and the cancer was the villain and moved into the nerve and kind of took over. It was a groundbreaking paper by Gustavo Ayala about 20 years ago that showed that there's this very dynamic interaction between nerve and cancer that actually occurs in this whole process. So subsequently, Magnin from Paul Frenet's group showed that really the sympathetic nervous system can be involved in the early stages of tumor development, in this case, prostate cancer. And then the parasympathetic nervous system can be involved in the later stages when the tumor proliferates and spreads. Other studies have shown similar effects in pancreatic cancer, for example, as well as in gastric and basal cell carcinoma, where they've shown that denervation basically suppresses carcinogenesis. So the mode of origin and progression seems to vary to some extent from one cancer to another. In today's talk, what I'm going to tell you about is some mechanistic studies that we did, and then went on to explore the clinical relevance, and finally talk about spatial transcriptomic analysis, which I think is what many people here are very interested in. So this mechanistic study begins with this protein called gallinin receptor 2, which is a G protein coupled receptor. There are three forms identified: gallin receptors 1, 2, and 3. And as is typical for G protein coupled receptors, these have this 7 transmembrane domains. These receptors have multiple effects in both neuronal and non-neuronal tissues. And we first got interested in this was because gallin receptor 1 was shown to be deleted by Tom Carey's group, showed it to be deleted in about 75% of head and neck cancers. And we went on to do some signaling studies in that and found out that while gallin receptor 1 acts as a tumor suppressor, another member from that group, gallin receptor 2, seems to have an antagonistic role with gallin receptor 1. So gallin receptor 2 has more of an oncogenic role. It can either be overexpressed or you have removal of the tumor suppressor, which in essence amplifies its signaling mechanism. And we characterize this in a series of papers over the years. Now looking at gallin receptor 2 in the context, we talked about it in terms of proliferation, tumor growth, and survival. We looked at it in the context of invasion, which is a critical oncogenic phenotype. And here we're doing standard Matrigel assays where you look at the migration or movement of the cells through the Matrigel with the intent that the Matrigel kind of simulates the connective tissue that underlies the epithelium in the oral cavity. And what we found is that overexpression of gallin receptor 2 in oral cancer promotes the invasion. And when you knock out the ligand for this receptor, the ligand being gallinin, you get a suppression of this invasive phenotype. So moving this into an in vivo situation, we find here, we use the chick egg, and the reason we use the chick egg is because it has this chorioallantoid membrane, which is the chorionic epithelium, separated from the underlying connective tissue by this basal membrane, which consists of type 4 collagen. And that's similar to the collagen present in the oral mucosa. We learned this model from Steve Wise over at LSI, and essentially you can implant GFP labeled cancer cells and then look at tumor growth, study invasion, angiogenesis, as well as metastasis in this model system. So looking at that, what we see over here is when gallin receptor 2 is overexpressed, you have this disruption, or we have this disruption of the collagen that would highlight the basal membrane region of the epithelium, consistent with a more invasive phenotype, and you can actually count these islands and determine and quantify invasion. More importantly, we looked at metastasis and found that when receptor 2 is overexpressed, it dramatically promotes metastasis to the lower cam and to the liver in these cells. Now, gallinin, the ligand of this receptor, was actually originally identified as a neuropeptide. And so we looked at gallinin as well as some other neuropeptides and neurotrophins in the context of head and neck cancer in multiple studies, and found that gallinin is highly expressed in head and neck cancer relative to corresponding normal tissue. And this contrasts, for example, with nerve or neurogrowth factor, nerve growth factor, which is often studied in the context of neural invasion. Additionally, we observed that gallinin actually, gallinin expression correlates with poor survival, and this is at 3 years, as early as 3 years, and once again, contrasting it with NGF in these same tumors. So this seemed kind of interesting to look at in terms of oral cancer, and the fact that it was a ligand was a neuropeptide prompted us to look at its interaction with cancer cells. So what we did over here was take 2 different cell lines. SCC1 is a GFP labeled oral cancer cell line, and SH-Sci5Y is a neuronal cell line that we used. And then we co-cultured them And we looked at the effects of SCC1 when it had an empty vector versus overexpressed gallin receptor 2. And co-culturing neuronal cells with oral cancer cells overexpressing gallin receptor 2 led to the differentiation, and that's seen by this neurite outgrowth, led to the differentiation as well as co-localization of these two cells. To verify that it was the condition medium factors released by the cancer cells that were modulating this whole phenotype, we collected condition medium from control or gallin receptor 2 overexpressing oral cancer cells, place that condition medium on neuronal cells, and see again that very dramatic differentiation. Subsequently, we went to look at the interaction using dorsal root ganglia as a in vitro model, and then the cancer cells. And you can see when gallin receptor 2 is overexpressed in these cancer cells, there's this great attraction towards each other between the nerve and the receptor. Downregulation of gallinin in these conditions using a gallinin antibody abrogated this effect. And then we could also abrogate this effect by using a GALA2 biochemical inhibitor, M871. And when we see the introduction of the inhibitor, we block that effect over there. The scrambled peptide uses control for this small molecule inhibitor had no effect in contrast to that. So at this point now we wanted to look, our real goal was to look at this interaction between the nerve and invasion. So we used a DRG CAM model. And in this case, what we do is we implant, we retrieve rat DRGs, dorsal root ganglia, and we graft them onto the CAM membrane and then can study this interaction. And to cut to the chase here, what we basically see is when you have overexpression of gallin receptor 2 in cancer, you have this very great attraction between the cancer cells and the neural tissue. And we can measure this invasion, cancer invasion, as well as this neurite extension. So ultimately, the bottom line is that the cancer and the nerve have this very dynamic interaction between each other. And this gallin receptor 2 induced pathway is very important for perineural invasion. And we went on to characterize this mechanism. It's been published, so I'm not going to go into detail today. But what I want to emphasize is that what we observed really is that this biochemical interaction between cancer and the nerve appears to occur prior to physical contact. Now, subsequently, other groups showed that in the context of oral cancer, the neural system has a significant role. This is Jeff Meyer's group that showed that loss of P53 actually reprograms those cells into and allows this invasion. And more recently, this effect of gallin was characterized that's occurring via the Schwann cells in these tumors. Okay, so we know that this has a role. So what's the clinical relevance of these findings? And to understand this, let me take you a little bit through this whole definition of perineural invasion. So as I mentioned earlier, the tumor has to be completely surrounding or at least surround 33% of the nerve or be present within the nerve itself. That fulfills the current criteria that we use clinically when we diagnose cases of cancer and whether there's perineural invasion involved. So it's a close proximity to nerve and involving at least 33% of its circumference. But then you look at cases like this, and in essence, since the nerve is not up against the cancer, or in this case, the nerve has less than 33% of cancer adjacent to it, these would be called PNI negative. But I just showed you how in vivo, when we look at it in a mouse model, or excuse me, a CAM model, the nerve and cancer have this very great attraction to each other even before they're physically in contact with them. So are we underdiagnosing these cases? And why does this, should it be redefined? And why is it necessary to redefine this. As I mentioned earlier, PNI is an adverse pathologic feature in multiple cancers. Patients with PNI receive aggressive treatment, but the diagnostic criteria we are using are archaic and based on criteria established clinically over 40 years ago. And it doesn't take into to account the recent biologic mechanisms that have been identified. And the other question we want to know is, can PNI be an independent predictor of prognosis? As I mentioned, it's often linked to lymph node involvement and used as a surrogate for lymph node. Can it be done independently? So in order to answer these questions, we looked at tissue from patients, from 71 patients from the head and neck oncology program, formerly the head and neck SPO. And these are all oral cavity squamous cell carcinomas. And this tissue is important because it's linked to clinical outcome and is sent to us totally de-identified. And then once we finish our analysis, we send it over to cancer biostatisticians, specifically Jeremy Taylor and Emily Belisle, who do a phenomenal job with analyzing it. So this was our pilot study. And I'll cut to the chase of the pilot study because that's published. And what we see here is that this is not an all or none phenomenon. Basically, as the nerve tumor distance decreases, then the survival of these patients improves. So what I'm going to talk about now is all unpublished data, and it is in this context that I talked to you about the transcriptomics. So subsequent to that pilot study, we did a validation study with an independent cohort of 71 patients. The data we got is very similar. So what I'm going to present to you today is actually a combined analysis of all 142 patients, all with oral cavity cancer. This is looking at formalin-fixed paraffin-embedded tissue. We did stains on this and then did quantification to determine patient-level data, as well as what we call nerve-level data. That means each nerve was analyzed. And subsequent to that, we use these subset of these specimens to do digital spatial profiling of these tumors. So give you a brief overview. The first column is cohort 1, the second is cohort 2, and then the combined cohort over here. And what we basically can see is we have about an equal gender distribution. This is a little atypical. Typically, oral cancer is a disease of more males than females, but we have an equal gender distribution. The oral cavity subsites was primarily the tongue, which is pretty classic for oral cancer. And then we did several stains in these. We did a hematoxin, eosin, cytokeratin to highlight the cancer, S100, TUJ1, and GAP43 were all to highlight the nerves for different purposes, as will become apparent later, and then an IgG control for each one. And what we did was, and this was work done by Nicole Russo and subsequently by Lydia Schmidt and Cindy Perez, graduate student and postdoc in the lab. And essentially what they did was quantify the distance between every nerve within what we call the tumor bulk and the adjacent tumor island, extending 2 millimeters out from the invasive front of the tumor. And what we found was when you assess PNI on H&E alone, that is actually an underassessment compared to combining the H&E stains, the S100 and TUJ1 stain that I showed you, with the cytokeratin, and we actually have an increased detection of perineural invasion in these cancers. Keep in mind that most of the time, under clinical circumstances, people are just using the H&E. So subsequently, the data I'm going to show you is from this combined detection. Okay, so one of the first things we observed was that perineural invasion or cancers that were PNI positive had were basically had a worse prognosis, and this is using disease-specific survival. So this is the most stringent survival that we could look at, and essentially what that means is the patient died of their disease. So here we have with patients who are PNI positive, have a poorer survival, and this is supports that it is actually an independent predictor of poor prognosis. Now we compare this with the end stage. End stage is the lymph node, which is lymph node involvement is used as part of the staging system for these tumors. And as I mentioned earlier, that staging system is relevant when treatment is selected. So as is known already, patients with positive lymph nodes have a poorer prognosis. So now we want to tease apart this connection between patients who don't have lymph node involvement. What is their connection to PNI positivity? And so taking these N0 patients and dividing them into those that were N0 and PNI negative and N0 and PNI positive, what we see over here is that the lymph node negative patients who have perineural invasion actually have poor survival similar to those who are lymph node positive. So this was a very important determination. And here we look at the hazard ratio. Just PNI positivity indicates a poorer survival. But now when you look at PNI positivity in the context of negative lymph nodes, you see this is associated with a high hazard ratio. Again, these orange colors indicate significance. So now the next thing we determined, which is what we were trying to find out from the start, was what is this difference between nerve tumor distance? Does it make an impact on the survival of the patients? And here we look at 27 microns. So how do you come up with 27 microns? That's the statisticians using regression tree methods to separate the samples, or the patients, into two groups based on disease-specific survival as and looking at distance as a covariate. So when we see this, patients with a distance, nerve tumor distance of less than 27 microns have a greater propensity to die or a poorer survival than those who have greater than 27 microns. Now separating the PNI, so this includes everybody, right? But there are some in this group of less than 27 microns that would already be considered PNI positive. So we separated the PNI positive and the PNI negative, who now have less than 27 microns or more than 27 microns. And these patients would normally be considered PNI negative, but we can see that when they have a less than 27 micron distance between the nerve and the tumor, they actually behave as poorly as if they were PNI, or almost as poorly as if they were PNI positive. And once again, looking at this, the greater the nerve tumor distance, the better the survival of these patients. Okay, so using a Cox generalized additive model, and this is including all the nerves at this point. So from 142 patients, Leja and Cindy measured 9,127 nerve tumor distances. And essentially what we see is, as I showed you quite early on from the pilot study, that the death rate, the disease-specific death rate of these patients decreases gradually and plateaus at about 500 microns. So this supports that the distance between the nerve and tumor is relevant in terms of whether it's close or touching. So the touching would be if this distance was 0, but you can see this gradual decrease in the death rate, supporting that the distance is important till about 500 microns. And this, looking at this in N0 patients, once again, we see that the distance less than 27 microns is important for survival of these patients. Okay, so now we turn our attention to the nerve diameter. And this is important because there's been kind of discordant studies, if you will, talking about the involvement of nerves and the width of the nerves within the tumor specimen. So we looked at nerves within the tumor bulk, that's within the area of the tumor, and classified them into small, medium, and large based on tertiles. And the larger nerve diameter is associated with poor disease-specific survival. Once again, using regression tree methods, you can see a diameter more than 32 microns is associated with poorer survival. Now, why is this relevant? What's relevant is there's a study in 2005 that published that nerves over 1 millimeter are relevant. If PNI occurs in nerves over 1 millimeter, then that is associated with poor prognosis. Now, in all these 142 patients that we looked at, nerves greater than 1 millimeter in oral cavity cancers were observed in less than 5 patients. So 1 millimeter is not a great cutoff to use to try and assess the involvement in oral cavity cancer. And so that's why we reassess this in the context of this regression tree methods. And once again, looking at the hazard ratio, the greater the diameter of the nerve, the poorer the disease-specific survival. Okay, so now we've looked at it, the diameter, we did the Cox model again to assess the association of disease-specific death rate with the nerve diameter. And you can see as the nerve diameter goes to about increases, there's this increase in the death rate, and then it falls off. And we think at that point, they're very far from the tumor itself. Nerve diameter associated with N0 or node negative patients, if the nerve diameter is greater than 32 microns, we see this association with poorer survival. So from these 142 patients, what I've shown you is how there's this association with poor survival, both at the patient level and then at the nerve level. We've dissected basically the parameters in this particular case, the nerve tumor distance as well as the diameter of the nerve. Now we want to find out whether this clinical study is rooted in some transcriptomic changes. So our hypothesis is that these nerves that are closer to the tumor will exhibit a different transcriptomic profile from nerves that are distant from the tumor if they are active participants in this nerve tumor interaction. And to understand this, we did digital spatial analysis on a subset of these patients, and we selected 8. All are from tongue tumors to avoid any differences from site specificity. And essentially what we did here was we stained these with cytokeratin and S100. And I have to tell you that this is the first time we were doing these spatial transcriptomics. And the core at the NCRC, Olivia Coos and her group were extremely helpful in this whole process. And this was work done by Legia. And then we selected regions of interest, and then finally went through the analysis, which was done by Weixing Wu also over at the core. And each of these circles over here, firstly, the purple staining over here is the tumor. So the cytokeratin highlighting the tumor. And this greenish staining are the nerves highlighted by an S100 stain. Each of these circles represents data from areas of interest. So these are the 4 main areas of interest. And we investigated nerves close to the tumor, nerves at a distance of 100 microns, then and sequentially at 1,000, and then greater than 1,000, which we called nerves far from the tumor. And then these circular plots in red show genes that are downregulated, in blue show genes that are upregulated in the context of nerves close to the tumor. And these panels over here are just a segment showing it at a linear level. So when we look at this GeoMean, which is the log2 transformed normalized gene counts, what we see very clearly is that the nerves close to the tumor have a downregulation of genes, and this change is gradually abolished as one gets to the nerves far from the tumor. And the same thing is observed in the upregulated genes. It's a gradual transition, except when you compare the nerves close to the nerves far, you can see that is very dramatic, even when with the adjusted p-value. So in essence, the transcriptome changes gradually with the nerve tumor distance. The number of genes that were differentially expressed in NC, or nerves close, was 159. 95 of these were upregulated and 64 were suppressed. So what are these genes? Okay. Yeah, so what are these genes over here? Here's a heat map, and these little pink arrows highlight the genes of interest. Basically, these were genes related to protein production, neurodegeneration, nerve growth, and response to stress. So for example, here we have myelin basic protein that is downregulated in nerves close to cancer. And myelin essentially in the nerves facilitates electrical conduction. That's how the nerves transmit information. And when myelin is lost, and it's lost in some neurodegenerative conditions as well, the transmission of information cannot occur very well. And in sensory nerves in the oral cavity, myelin can be damaged when there's inflammation or irritation to the tissues. PTPRZ1 has not been really shown in the peripheral nervous system or studied much in the peripheral nervous system, but in the central nervous system, it's associated with remyelination, and you can see it's suppressed in these conditions. MARX, which is myristylated alanine-rich C kinase substrate, cofilin 1, and axin A2 are all upregulated, and these are associated with nerve regeneration. And then ATF4, GSTP1, and FAB5, to name a few, are associated with the stress response in these tumors. Now, the nerves, I mentioned that we looked at the nerves, and the reality is that because this is not single cell sequencing, we cannot differentiate between the actual neuronal cells, or the neurites, and the Schwann cells, because that's all highlighted in the nerve. And so to validate the findings that we got from the spatial transcriptomics, we actually looked at expression in the neuronal cells and Schwann cells. And this is a time course of expression of some of these candidates associated with either nerve regeneration or stress or myelin, I'll show you in a minute, and essentially at different time points. And what we see over here is that all these are significant at one or another time point. And when we look at the Schwann cells, the one that I really want to highlight here is this dramatic drop-off in myelin basic protein. So what are we doing here? We are looking at whether the tumor cells secrete a factor that affect the neuronal and Schwann cells. So we isolate conditioned medium from tumor cells and then treat these neuronal or Schwann cells for these varying periods of time, and then assess expression of these different transcripts that came up on the spatial analysis. So myelin basic protein, as I mentioned, is downregulated in these nerves that close to cancer. And this is consistent with a demyelination or injury response. And so we verified that on tissue specimens from these patients and essentially scored it as weak, moderate, or strong, and then the stain proportion as a continuum over year. And then we did a combined score, and what we see is that in nerves close to the tumor have a lower score than nerves at a distance, suggesting or supporting that injury is occurring in these nerves that we saw also in the previous slide when we did the functional or the assays in the in vitro assays. And then finally, what we looked at was this GAP43. You may recall earlier I said it was one of the neuronal stains we did. And the reason we used GAP43 is it measures neurite outgrowth, so axonogenesis. S100 and TUJ1 will highlight the entire nerve. GAP43 will highlight new nerves or neurite outgrowth. And this is interesting. So what we have over here is we looked at GAP43 expression relative to TUJ1, which highlights the entire nerve. And we scored these as either staining less, equal to TUJ1, or greater than TUJ1 in these nerves. If it's staining more, one would expect more neurite outgrowth. And what we have over here is in the greater the distance between the nerve and the tumor, the less GAP43 staining you have, so the less neurite outgrowth, which was to be expected. So to summarize what I've just told you, the transcriptomic changes in the nerves vary with the nerve tumor distance. And this correlates with disease-specific survival. The tumor cells themselves secrete factors that induce this injury response in the nerves, and axonogenesis or neurite outgrowth occurs close to the tumor. So in essence, what we find is that nerves close to tumor and large nerves are as important as what we currently call perineural invasion in predicting poor survival. And nerves close to the tumor undergo an injury-like degenerative process that may stimulate nerve growth to induce repair. This is the first study in humans to show nerves respond to tumors in an injury-like process and with striking correlations with clinical outcomes in these patients. So in essence, what we began as a diagnostic criteria and then went on to look at mechanism, and we are continuing to explore these mechanisms in data that I did not get a chance to show today. We found that those mechanistic studies prompted us to look at these clinical phenotypes that we have used for the last 40 or 50 years and to reevaluate them in the context of current biologic findings, and then do spatial transcriptomics, which supported that the current criteria need to be reevaluated, and that the nerves close to the tumor, the proximity to the nerve and tumor, is probably more important than how much of the nerve, etc., is involved. And then what we're hoping to do right now is we are working with a couple of people, Michelle Mirzwa and Keith Casper, who are radiation oncologists and ENT surgeons, and starting a clinical trial on tongue cancers. And we hope to include this as one of the parameters that will be investigated prospectively. And just to acknowledge the people who have done all the work, it's Cindy and Legia who've done the bulk of the clinical studies, some of the in vivo work done in the CAM model was by Min, and Nicole, who really started this whole staining for the clinical specimens several years ago. She was a research assistant in the lab, and Christy Scanlon, who was a dual degree DDS PhD student who did the mechanistic studies that I showed you earlier. None of this would be possible without excellent collaborators from the School of Public Health, Jeremy Taylor and Emily, who do an amazing job with the statistical analysis, and also from the School of Public Health, Laura Rozek, and from the med school, Gregory Wolf, who enabled us to get access to these tissue specimens so we could investigate the clinical relevance of these findings. And then Michelle Miaswa and Keith Casper, who helped a lot with the staging of these patients. The single cell sequencing core at the NCRC, I mentioned Olivia Coos before and her group, and Wishing Wu for all the analysis, and then grant funding from the Cancer Center core and then from the NIH. And I look forward to taking your questions and hearing your feedback. Thank you.
Thank you, Nisha, for that outstanding talk. Really marvelous work. Now the session is open for any questions. You can post it in chat window, or you could unmute yourself and ask. Maybe I can start with one question, Nisha.
Sure.
It's a very, very nice piece of work. I was just wondering how early the tumor cells disseminate into the nerve or underlying tissues. Like how many years before you would predict this process might be happening before you find cancerous lesions?
That's a really good question. So the couple of ways to think about this, and I'm going to stop sharing so I can see. So a couple of ways to think about this in terms of the tumor cell dissemination. It's very difficult to look at it in terms of the perineural invasion, because that could, in the oral cavity, because of the number of nerves in the oral cavity, this could happen amongst the early phases of cancer development. But I couldn't give you a specific time because we don't know that. Now, one of the things that I could say over here is there's something called perineural spread. That is detectable on radiographs. But in order for that to happen, the patients have to have pretty extensive neural involvement such that you could detect it. So perineural invasion would be an earlier phenotype to an earlier detection rather than perineural spread, because once perineural spread occurs, there's so much gross involvement of the nerve by tumor that the survival would my guess would nosedive even further. Now, another thing I wanted to mention is I mentioned a study that had looked at nerve diameters, and those studies have kind of looked at excision specimens. Everything I showed over here was done with incisional biopsies or with one section from an excision. And why that's relevant is because if you think of it in terms of treatment planning, you would want to know ahead of time whether you should use this as a surrogate marker, for example, lymph node removal. Right now, the only way to tell if there are occult mets in the lymph node is to actually remove the lymph node, which itself is an aggressive surgery independent of the oral cavity surgery. So that's why I think what we're trying to look at over here in incisional biopsies or in a single section from an excision is important.
Thank you very much. Any other questions, please?
Can I ask you a question?
Sure.
Yes, please go ahead.
Hey, Nisha, beautiful work. Congrats. So I have a question about the second part, the spatial transcriptome.
Yes.
So you showed that they're heterogeneous, or there's a difference, depends on the distance close to the tumor. So I may have missed that. Did you compare that to the samples without tumor? So I just wonder if the heterogeneity correlate to different kinds of nerves in the normal sample or normal condition, or this is more like tumor-induced response?
So that's a really good question. So the way we address that, remember I said we looked at nerves far from the tumor. So we purposely selected specimens that had nerves, what we call outside the tumor bulk. So those nerves would technically be uninvolved, what they would call the adjacent margin of the tumor, meaning the clear margin. And the reason we did that is we wanted to get nerves that were clear of tumor, that did not have any tumor, but yet were within the same patient, so the comparisons would be tighter than incidental findings, like a patient could have inflammation in the oral cavity that was unrelated and could have changes. So we compared nerves that were totally uninvolved with nerves that were involved in cancer. Now, to answer your question more directly, we have done studies in mouse where we have taken nerves from a totally independent mouse and taken nerves from the same mouse adjacent to the cancer and then contralaterally. And over there, when we look at nerves from a totally independent mouse, meaning without tumor, versus a mouse with tumor, but on the other side, so away from the tumor, we don't see differences. So we felt quite comfortable using the nerves far and nerves close as a comparison for the spatial analysis.
Thank you.
But you bring up a really good point.
Sure. Any other questions? I had one more.
Yes,
I was just
sure.
wondering about your studies, how relevant they are if it is not a oral cancer, but there is a neural invasion in other cancer like a prostate cancer. Your findings are still relevant? What are the parts that we could directly take it into other cancers versus which we could not?
So I think, firstly, I think, the criteria we used for perineural invasion when we did the clinical study, what we did was, because there's so much variation between cancers and how people call perineural invasion, what we did was we took the criteria from all cancers. We literally went through the literature and said, what do they call it in GI? What do they call it in prostate? And we evaluated all those parameters in that pilot study. I didn't show all that data because it was a lot of data, but it helped us cull out what were the most relevant. And then we took that and amplified it in the second study. Now, I do think that there can be a variation from cancer to cancer. And simple explanation would be some cancers might have more of a sensory component, others might have more of a sympathetic or parasympathetic driver in there. So in terms of the genes of interest, there may be a variation, but in terms of the phenotype, I think it's highly relevant to what we could be doing. In fact, the whole concept of perineural invasion was first defined in the context of head and neck cancer by Batsakis, 40 years ago. So, so I think there are parts that can be applied and there are parts that have to be cancer specific. Now, the spatial transcriptomics, no one has done that in any human cancer. So I think it opens up a field of investigation. What's really exciting is that we find it's like a regenerative phenotype that the cancer basically induces in the nerve, which from a cancer perspective is pretty smart to do because you allow yourself then to spread into this neural tissue.
Yeah, thank you.
You're welcome.
Any questions for Dr. De Silva?
Okay, yeah, I have one question. It's very fascinating story as usual. Okay, so so the distance plays a role, which may suggest something secreted from the nerve.
Yes.
Yeah, but it's still possible. There's a direct communication through axon to the nerve. Then if they become closer, the more frequent direct communications. So that would be another explanation. But how would you like to comment that?
So I just want to make sure I understand your question correctly. So you think the axons themselves could be directly interacting with the cancer?
Right.
Okay, and I agree with you. That's a possibility. Except when we did the in vivo studies in the egg, the axons and the cancer were not actually touching each other. So it was a factor. And then in order to also characterize that even further, we actually did studies where we would take the neuronal cells separate and collect the conditioned medium from them and then treat the cancer, or do the reverse, take the conditioned medium from the cancer and treat the neuronal cells. And we would see that difference. And in the in vivo studies, which I didn't get a chance to show today, if it's some of the mouse studies, we are seeing a similar effect with that, where we can actually characterize that it's a secretive factor. But I do think ultimately the axon outgrowth will affect progression of the cancer. But the cancer is actually inducing that axon outgrowth, and that whole process of disrupting the myelin actually encourages it to regenerate. So,
okay, great. Yeah, thank you.
Great question.
Any last minute questions?
If not, thanks everyone for attending. I really appreciate.
Yeah, thanks again. We thank you again, Nisha, for presenting in spatial analysis program. Thank you everyone for attending. Have a nice afternoon.
Bye.
Bye. Bye-bye.